Okay, so I think we're on stage now. So perhaps I can start with a few words. So hello, everyone. Thanks for joining us today. I'm really happy to see that the topic that we've chosen today has attracted such a large audience. So it's great. So this is the first event that we are organizing since we launched a few months ago with my colleagues Boris Chomet and Raoul Belzo, a new section that is dedicated to the role of epigenetics in psychiatric disorders. So this section is part of the French Association for Biological Psychiatry and Neuropsychopharmacology, the AFPBN. And so, of course, if you are interested in joining us or participating in some of our events, please do not hesitate to contact us. So for this first event today, what we wanted to do with Boris and Raoul is to try to organize a dialogue to discuss about two trends in current research on major depressive disorder and how the field should try to deal with the tensions that might be created between these two trends. So the first one refers to, as you all know, the recognition that psychiatric disorders, including depression, are very heterogeneous conditions that rely on complex interactions among multiple factors. And so to try to identify molecular markers that can be measured, for example, in peripheral blood tissues, and that could be helpful for diagnostic or therapeutic decisions, it is expected that very, very large cohorts will be necessary. The second trend, on the other hand, corresponds to studies that have been conducted on postmortem brain tissues at higher and higher resolution, ultimately in single cells, and probably also in the near future, looking at several types of molecular or epigenetic processes in each individual cells. And so to try to describe each of these two aspects, we'll have two talks today. So the first one is going to be by Dr. Chen Shui-Yi. So Chen is a research fellow at the University of Edinburgh, and her recent work has focused on epigenetic peripheral biomarkers of major depression, as you will see. The second talk, also during approximately 20 minutes, is going to be from Dr. Corinna Nagy. So Corinna is an assistant professor at McGill University at the McGill Group for Suicide Studies, where she's conducting single-cell studies on depression. And so after these two talks, we'll have some remaining time for questions and answers. So feel free to write your questions in the chat. And so I guess that's it. So without further ado, of course, I'd like to thank Chen and Corinna for taking the time to present their work. We are very happy to have you. And so, Chen, the floor is yours.
Bonjour à tous. Good afternoon, everyone. It's my great pleasure to be here today to present one of our recent publications, and it's a DNA methylene-wide association study of polygenic risk scores for depression. So first of all, I'd like to just give a very brief introduction of what DNA methylation is. We know that the genome includes more than just DNA sequence. The classic double helix structure shown on this figure represents the DNA sequence that we're familiar with. As indicated in the figure, numerous epigenetic markers are also tagged along to the DNA sequence. Epigenetic markers are really important biological components that influence the interpretation of DNA without changing the sequence. And one of the most studied epigenetic markers is called DNA methylation, as circled in the figure. In mammals, DNA methylation takes place by adding a methyl group to a cytosine followed by a guanine, which is called CpG, and that's where DNA methylation takes place. DNA methylation is known to be able to capture a lot of environmental risks that can be very relevant to mental health disorders, such as smoking, alcohol consumption, and BMI. Other than just environmental factors, DNA methylation is also associated with genetic variants. On the right-hand side, as you can see in the figure, it shows the results of a recent study looking at more than 1,000 pairs of twins. and they found that DNA methylation can be the vast majority of the CPGs receive influence from both genetics, indicated by the red bars, and also environment, indicated by the green bars. So next is our key question. We're interested in the link between MDD and DNA methylation. As we know, MDD is a heritable trait with a heritability of around 37%. And the most up-to-date polygenic risk scores explains around 2% of disease liability. And polygenic risk scores has been thought to be one of the most robust biological predictors for MDD. However, the relationship between genetic risk of MDD and DNA methylation has been very much understudied, and that is likely due to a lack of power for both genetic data and DNA methylation data. And DNA methylation data in specific, study sample size tends to be really small. A study often has a couple of hundred individuals or even less. And in our study, we conducted a methylome-wide association study using multiple large data sets. Here I'm just diving into a little bit of the analysis outline covered in this study. First of all, we conducted a discovery MWAS on Generation Scotland, followed by a replication MWAS on an adult sample consisting of Lothian birth cohorts and ALSPAC. These are adults, as well as a different replication on ALSPAC children. And finally, we used Mendelian randomization to investigate the causal relationship between DNA methylation and MDD. So first, the discovery MWAS conducted in Generation Scotland. As you can see, the sample size is way bigger than the previous studies that I introduced. We used data from over 8,000 participants in the Generation Scotland sample. Polygenic risk scores were created using the latest MDD GWAS. 9 P thresholds were selected for creating the polygenic risk scores, ranging from 1 to 5 times 10 to the minus 8. So for P threshold of 1 indicates all SNPs in the GWAS included for creating the genetic risk, and on the other extreme, 5 times 10 to minus 8, that's the genome-wide significance level, and only those top hits were used for creating polygenic risk scores. And for the DNA methylation data was indexed using M values on EPIC array. Covariates included smoking, age, sex, methylation, and genetic principal components, and also DNA methylation estimated cell proportions. So here is the first descriptive of the summary of the results. If you could follow me and look at the left-hand side of the panel, the x-axis represents the P threshold used for creating the genetic risk scores. From left to right, the P thresholds becomes more stringent. And the y-axis represents the variance explained by the polygenic risk scores predicting depression. And as you can see, as the P threshold becomes more stringent, variance explained becomes lower. And on the other hand, if you look at the right-hand side of the panel, for polygenic risk scores created using a more stringent threshold, the number of CPGs associated with them was higher on the right-hand side than those with a more lenient threshold on the left-hand side. So on the left panel, there's a downward trajectory, and on the right-hand side, there's an upward trajectory. As a follow-up, we then looked at the MWAS results for the polygenic risk scores created using only the genome-wide significant SNPs, which show the largest number of associations with DNA methylation. And here is the Manhattan plot. There are in total 70 CPGs associated with the polygenic risk score. And of all the significant CPGs, over 64% were in the MHC region in chromosome 6. And 25 CPGs were outside of the MHC region. And therefore, we conducted pathway analysis on those CPGs located outside of MHC region to find out the biological pathways associated with it. So here I'm showing the top 10 gene ontology terms associated with them. And most of them are, as you can see, related to neuronal and brain development, such as those relevant to postsynaptic density membrane, postsynaptic specialization membrane, and maturation in the cerebellum. I'd also to highlight that although we used data from whole blood, the top pathways we found were still brain-related. So moving on from the discovery MWAS to replication, first we looked at, first to give you a quick introduction of how we looked at the replication, separate analyses were conducted on adult sample of 2,049 participants, and another replication on samples of 423 subjects. DNA methylation data window.
Okay, so it seems we lost Chen. Chen, if you can hear me, perhaps you could shut off your camera, perhaps.
Right. I hope you can hear me.
Yeah, we hear you, but maybe you could shut off your camera to save.
That's great.
Yeah. Thanks.
Yeah. Thank you very much. I'm very sorry for the disturbance.
Okay.
Okay, I'll carry on. So I think I've gone to introducing the DNA methylation data. The last point is that we kept all the covariates consistent between the discovery and replication samples. Here's the results. As we can see, first we look at the MWAS results for polygenic risk scores across all P thresholds. And there's a similar trend found similar to the discovery analysis. The number of CPGs associated with polygenic risk scores created using a more stringent threshold was higher than those with a more lenient threshold. So you can again see this upward trajectory in this plot. And next, we focused on, again, the results on the MWAS for polygenic scores created using the genome-wide significance SNPs. In the scatter plot, as you can see in the top right corner, in the scatter plot, the X and Y axis each represent the effect sizes for the discovery and replication analysis. The effect sizes are highly correlated with or without CPGs located in the MHC region. And if we look at the same MWAS, rather than conducted on replication in adults, but rather in the replication in Alzheimer's children, effect sizes were again highly correlated if you focus on the scatter plot. To go one step further, we noticed that in the significant CPGs, around 12% of them are not close to or in LD with any of the genetic risk variants for MDD. In order to find out why, we conducted MWAS on each of the lead genetic risk variants. After clumping, there are 96 genetic risk variants included in the analysis. And here in this heat map plot shows the significant associations between CPGs and SNPs. The roles, they represent each individual SNPs included in the analysis, and the columns represent the CPGs. Any association that are circled out in the diagonal line are local associations, and those associations of the diagonal line represent distal associations. And you can see a lot of the associations are actually distal, and that consists of 87% of the genetic variants showing distal effects on CPG sites. In particular, I'd like to point out the 5 different SNPs that showed very widespread trans effects across the autosome, more than half of the autosomes. So this result here indicates that there might be some key variants driving many of the distal associations. Finally, we used Mendelian randomization to investigate causal relationship between MDD and DNA methylation. For those who are not particularly familiar with the method Mendelian randomization, it is a genetic epidemiology method that can help us make causal inferences. It is often compared with randomized control trials. For those clinicians on the call, must be really familiar with this method. In a randomized control trial, participants are usually randomly allocated into 2 groups. One group receives treatment, and the other group is a control group and does not receive treatment. Later on, people would analyze whether the treatment group has a significantly improved disease outcome. If it does, then it proves that the treatment is effective. The logic of Mendelian randomization is very similar. The biggest difference is how the participants are allocated. Our genes, which are passed down randomly from generation to generation, can influence all kinds of factors, and that knowledge can be used to randomize people into separate groups. For example, if we want to know whether CRP is causal to systolic blood pressure, in a randomized control trial, we would compare participants who take CRP lowering medication against those who are randomly assigned to placebo. But in a Mendelian randomization study, randomization becomes comparing participants with and without genetic variants associated with lower CRP levels. And Mendelian randomization is particularly useful when it is impractical or unethical to manipulate the causal factor. And this is exactly the case for our study, because DNA methylation is very impractical to change in a living human being. So going back to our study, GWAS summary statistics were used for Mendelian randomization. MQTL data, which is genetic risk variance for methylation data, we used 2 sets of data sets. First is GoDMC as the discovery set, and then Generation Scotland as the replication set. MDD-GWAS, again, we used the latest PGC MDD-GWAS. For the CPG probes, we selected those probes with more than 5 genetic instruments after clumping, and those CPGs that were previously found associated with polygenic risk scores. And the MR methods we used were IVW, weighted medium, and MR-EGR. We hope that we can find consistent findings across all methods. And finally, the analysis went from the discovery Mendelian randomization using GoDMC, looking at the causal effect from DNA methylation to MDD. And second, we conducted a replication and bidirectional Mendelian randomization using dataset from Generation Scotland. And here is the results for the causal tests using Mendelian randomization. I'd like to draw your attention, first of all, on the left-hand side of the panel. The x-axis represents the log transformed p-values. A higher value would indicate a higher level of significance. And the y-axis represents each individual CpG tested. The red dashed line shown in the figure is the threshold for statistical significance. And here we can see many of the CpGs tested show the causal effect on MDD consistent across all three different Mendelian randomization methods. And next, I would like you to focus on the middle panel and especially compare the replication results with the discovery. You can see a very consistent pattern between discovery and replication analysis. And finally, on the right-hand side of the panel, there are some evidence for the reversed effects from MDD to DNA methylation. But note that the scale for the x-axis is about 8-fold smaller than the other two panels. So if we look at the significance statistics, the evidence was much stronger for the effect from DNA methylation to MDD rather than the other way around. And genes mapped to those causal CpG sites are involved in transcriptional activities, signaling receptor binding in the brain, and hormonal regulations. So in summary, we found very widespread replicable associations between polygenic risk scores for MDD and DNA methylation. And the second takeaway information that we have is that there are many genetic risk variants show the trans effects on DNA methylation, and 5 of them showed quite global effects across more than half of the autosomes. And thirdly, the associations were not just replicated in adults, but also found in adolescents at around the age of 17 years old. And finally, mutual effects were found between DNA methylation and MDD, but there's a stronger causal effects from DNA methylation to MDD rather than the other way around. And this finding is highly resonant with the adolescent MWAS. Taking together the MR results, I think DNA methylation, especially the causal and the age replication, indicate that there might be new ways of looking at DNA methylation for intervention. So thank you, and I'll pass it to Karina.
Hi, everyone. Okay, I guess those are the acknowledgements for Shen, and thank you very much for that great introduction to this topic. So I'm going to take you on a bit of a different track where we're going to zoom in much more resolved at single cells and see what they tell us about depression. So I won't go into much detail about the etiology of major depression or these epidemiological studies that show its influence on the general population. I'm sure everybody knows this already, but one thing I do want to point out is that there are many systems that are influenced, both centrally but also systemically, and that there are many causes that are compounded together. And I think that Shen showed that there are some interesting relationships between factors that might be influenced by these this composition of factors. The other thing I want to point out is that the prevalence and manifestation of major depression is quite different amongst females and males. So the disorder manifests differently amongst females, and it also seems to be very consistently across different populations, different geographic locations, has much higher prevalence in females. In terms of the neurobiology of major depression, and I just want to bring this into context because it will be relevant for the area that we chose to study. It really is focused in on the limbic regions of the brain, which have interconnectivity between the cortical regions and the subcortical regions. And so a lot of our molecular studies have focused in specifically on these regions so that we can actually be at the center of where these functional genomic changes are taking place. There has been quite a lot of research that shows, even at macroscopic level, so imaging, that these regions will function differently in the context of stress and in the context of stress in an individual who has depression. So, for example, this recent study shows that the reactivity to stress is actually different at baseline between males and females. So, in the prefrontal cortex, and that if you look at the sort of deactivation of the stress response in healthy females, it actually deactivates more rapidly than in males, but there is also a difference between the deactivation in these regions. So, for example, the amygdala, hippocampus, and nucleus accumbens all show a different level of deactivation in the context of healthy controls and MDD, and that there is also a sex difference in this. At the molecular level, we see that a relatively recent study looked at all of these limbic regions again. So to reiterate, cortical regions, ventral medial prefrontal cortex, dorsolateral prefrontal cortex, nucleus accumbens, and hippocampal regions are all showing some pretty stark differences in the gene expression changes that occur in the context of major depression. So when you take these changes, and we don't consider a threshold, but we really just rank them. So all the gene expression changes that occur in these different regions in males and all the gene expression changes that occur in females, and we look at them, then we see that in everything from the ventral prefrontal cortex, interior insula, nucleus accumbens, and ventral subiculum, which is part of the hippocampus, are extremely discordant, suggesting that there isn't actually a very big overlap in the dysregulation, also suggesting that this functional genomic change is extremely important between males and females. And a further meta-analysis of these data and other data that had been generated in bulk, so in bulk means at the level of all the tissue together, we see that there's a large discordance between males and females and how their genes are expressed differently in the context of major depression. And some of the theories that are behind this is that each cell or cell type is actually starting at a different start point. And one of the arguments that has been made by Sine and others is that certain cell types, for example, microglia, are starting at a very different transcriptomic start point than for males than females. And the changes that occur have to occur differently to come to the same endpoint. So all these transitional changes that we're seeing in these RRHO plots, these colorful histograms here, are to some extent the result of a change to arrive at the same endpoint, but starting from the same from a different starting point. So that kind of leads us into the idea that perhaps we need to look at these data at a much more refined level. If there are individual cell types that are starting at different start points, then in order to really understand what's happening throughout the development of the illness, we need to look at each of these individual cell types in isolation. And of course, there are many, many different cell types in the brain. So they're acting in concert with each other in their microenvironment, but they're also generating their own specific transcriptomic profile and epigenomic profile. And there have been to date many studies that have looked at bulk tissue. So again, using the homogenate of a piece of brain and looking at the effects of gene expression changes in this context and still being able to pull out signals from these individual cell types. But those signals tend to be diluted somewhat, especially if those signals are coming from a cell type that has less of an abundance in that particular brain region. Nonetheless, all of these there's been several studies that have shown, specific changes in specific cell types, or at least associated back to those specific cell types using marker genes. But typically what we get when we look at bulk is sort of a smoothie worth of information. So there may be many different individual pieces going into this output, but what we tend to get as a readout is the mixture of all of these. And if for some reason the composition of the cells are skewed toward one or another, so in this example, perhaps we're skewing toward red berries, then the flavor of the smoothie that you're going to get from this is red berries. It's going to hijack the signal, and we may have some information that is a little bit misleading, or that we can't really understand fully in the context of all the other cells that go into the information. And of course, to date, we've tried to in silico or analytically tease these things apart. But realistically, what we get is a breakdown of composition, but we don't get an exact signal. And then we can't compare those signals within the cell types. So the previous studies have done that. We get certain signals that we kind of probe directly for, but they can be very much skewed by the composition of the tissue that we're working with. So that's why we've moved to doing these single cell type studies. And we're doing this in postmortem tissue that we get from the Douglas Belgrano Brain Bank. This is a very rich brain bank, one of the oldest in Canada. If not the oldest in Canada. We have over 3,500 brains. And what's particularly special about these brains is that we do a psychiatric or psychological autopsy on the brains, which means that we go in after the individual has died, ask the next of kin a series of questions and demographic information. And we get sort of a background history on all of these brains, which is very important. It means that we can get an understanding of the type of depressive pathologies that they had. And we also can get other information. For example, if these individuals had early life histories of abuse or other adversities. So here's our first study that we did. It was kind of a pilot, if you will, to see how well this technology could work in postmortem brain tissue. And we went back to one of the most implicated brain regions, which is the prefrontal cortical region, and we used equal numbers of cases and controls. And this study was done to start in males. And I will get to why we did, well, essentially we did that because it was a pilot, but I want you to keep in mind nonetheless that we're very aware that the differences in psychopathology between males and females for major depression is very relevant. So if you can see this small cluster of nuclei, well, that's exactly what we had to do. We took the tissue and we broke it down into its elemental pieces. And normally we'd like to get cells, but because these brains have been frozen for, in some cases, 20 plus years, when we saw the brain, the cells lysed, so we're left with the nuclei. Luckily, the nuclei has an expression profile that is very reflective of the entire cell. And after we have each individual nuclei in suspension, we can use microfluidics to actually capture each individual nuclei in an emulsion bubble. So this will require that there's a coincidence reaction where we have an emulsion, we have a nuclei, and we have a bead. And the bead has many, many millions of barcodes on it. And those barcodes can then label each individual transcript of that given cell, or nuclei in this case, with a cell-specific barcode. After that, we then have all the information we need to analytically tease apart which transcript came from which cell. And that's exactly what we do as you move through this schematic, and you see this little square with all the dots on it. Those dots represent individual clusters, and those clusters are formed by assessing the entire genetic profile, or I should say genomic profile here, so gene expression profile of a given cell. And we compare that profile, that molecular profile of one cell to another cell. And we do this, as many times as it is required. So in this case, we have, this study had 80,000 nuclei that we compared. So each individual cell profile is compared to another, and those that are closest in their profiles are clustered together, and those that are more different are clustered away. And this allows us to then annotate the cell types based on marker genes. And then we can go in and do our further assessments, such as differential gene expression assessments. We can do network assessments, and then we can also follow it up with validation. And this is exactly what we did. So here you see this dot plot, And so each dot represents the enrichment of a given transcript. So these are marker genes on the x-axis, and these are the cell types on the y-axis. They are clustered by excitatory. We found 10 different excitatory cells. We found 7 different inhibitory types of cells. We found 3 oligodendrocytic cell types, and then 2 early oligodendrocytic cell types, which means oligodendrocytic precursor cells. And they all sort of cluster based on these, based on their profiles, and then we're able to sort of post hoc label what kind of cells they are based on these marker genes that are only expressed in those particular cell types. And then from there, once we have these clusters and their annotations, we can go in and look at the differential expression between the cell types within these clusters. And when we did this, we found that there were 96 differentially regulated genes. And this low number of differential regulated genes is because we had a very stringent cutoff here. We wanted to make sure that we were finding really the most salient findings, and it's very possible that we have a lot of false negatives. Nonetheless, we found that there were 2 clusters that seemed to be particularly dysregulated, and this was the OPC cluster. Okay, so going back to this early oligodendrocyte precursor cell cluster, so these are cells that are supposedly fated to becoming myelinating cells. And then excitatory 7 cluster. So this was a very specific, so not just excitatory cells, but excitatory cells that we could localize to the deep layers of the cortex. And then when we looked at the differentially expressed genes, we found an enrichment for these genes when looking in a database that associates all the genes that had previously been associated with different diseases. So for example, this one, we looked for psychiatric diseases, and the highest association between our genes and the genes that were associated with MDD came out as an enrichment. The other thing that we wanted to do is then look at how these cells might be communicating. And I think this is really important because we've kind of breaking these cells down to their elemental levels. And now we want to see within their microenvironment, is there something about the way these cells are communicating with each other that might lead to sort of this behavior and pathology. And so, of course, we wanted to focus in on the cells that we saw the most dysregulation in, which is this deep layer excitatory cell, and into the early oligodendrocytic cell types. And what we did here is, if you can look at the ligands, these are the signaling molecules that are coming from the excitatory cells, and then the receptors that are in the oligodendrocyte cells. And everything in blue are things that are either increased or present only in controls, and things in red are in cases or increased in cases. And you can the takeaway here is that you see far more signaling from the deep layer excitatory cells to the OPCs in the control condition, far less. So the communication is reduced in cases. And then if we look at it in the inverse manner, and we look at the signaling from the OPCs to the excitatory cells, we also see that there's far less signaling, and that one of the most influenced signaling pathways is the FGF pathway, which is actually fits with some of the recent literature that suggests that these OPCs actually may not be just destined to go through to myelinating oligodendrocytes, but actually have a modulatory effect on excitatory cells. And if you disrupt them in the context of depression, you actually do get depressive phenotypes in animals. So then we basically wanted to do a follow-up. We developed this model, and we wanted to sort of pick at certain molecules that we could then validate in situ. And we did this using RNAscope. And in terms of, so we found something that we were able to localize changes to excitatory cells in deep layers. But in order to do that in situ, we then also needed a marker that represented this deep layer excitatory cell type. So we used RFXP1, and we co-labeled that with the transcript of interest, and we actually were able to recapitulate the changes that we saw at this high throughput sequencing level with this low throughput in situ molecular counting level. And we were able to show these changes in the excitatory cells as well as the OPCs. So as I mentioned at the very beginning, there are vast differences that have been noted in the literature between males and females. And we've also seen molecular studies that really seem to show that the gene expression profiles and the way they change in males and females in the context of depression seem to be quite different. So it made perfect sense to want to follow this up and expand our cohort with females. So we went from this smaller cohort of 34 males to add another 40 females, and we had to drop some, but anyways, we have a total representation here of 71 samples that we were able to look at all together. And so we processed more nuclei from the females exactly in the same way that we did for the males, and we were able to generate these much larger this is a t-SNE plot, which just shows that each individual dot represents a cell, and this is how they cluster together. The colors represent these broad clusters that we were able to form, where the largest cluster here is excitatory. And because we combined these data with the previously generated data, this made a nice, very large data set of about 180,000 nuclei that we were able to sort of look at in the context of depression. And the upper t-SNE shows broad clustering. This lower t-SNE is actually showing the way that we can really get fine cluster labels in the way we choose to do the clustering. So basically, we alter the parameters such that we can say this cell type is different enough in terms of their expression profiles that they actually represent a different cell type or state. And then we also were able to once again label them in the context of their layering in the cortex. So this is actually just a subset of the clusters, and these are just the excitatory clusters, and these are how we were able to annotate them to the specific layers. And this is, so when we first did the male data set, there were very few others who had actually done any single cell work in the human brain, and now there are many more studies, so we can actually leverage those studies to make sure that we have consistent findings and annotations, and that's exactly what we did. This lower t-SNE plot, or UMAP, is actually a UMAP that overlays our data with annotations that were generated from technologies that allow us to do the transcriptomics in situ, so actually have spatially resolved transcriptomics. We overlaid that, and we were able to see that our annotations of layer-specific excitatory markers were quite accurate. We're also able to leverage much larger data sets that have been done in the brain at a different sort of control level. So there are many, many brain initiatives now that are trying to look at individual cells in the brain and really classify them, characterize them. And the Allen Institute is leading the charge on this. And so we were able to leverage their data to make sure that our annotations made sense. So now with the influx of all this data, we have a lot of checks and balances and sort of are able to validate that what we did previously holds still. And so the biggest question that we had to ask once we did all our annotations, all our clustering, is what do the differences between males and females in the context of depression look like? Is it similar to what we have seen in bulk, where there are just stark differences in the levels of gene expression, or is this maybe something that we couldn't pick up correctly in bulk? And I think that our results suggest that by and large, the changes, when we rank them, same way that it was done by the Nestler group, when we rank them by, most increased and then to the most decreased, we're by and large seeing concordance. So I think that that's somewhat reassuring, that males and females aren't completely different, and we are all humans, so it makes sense. And that the changes are those that don't pass the significance threshold are really occurring in a very like manner. Now, that being said, there are still some clusters that seem to show discordance. So this might suggest that there are certain cell types that are really responding in a very different way. But I do want to emphasize that in general, we are seeing that the changes that are occurring are by and large occurring in a concordant manner. Now, if we take the differential expression and we want to look at the differential expression in a thresholded way, so we really look at what rises to the top as being extremely changed, then here is where we're seeing changes that are quite different between males and females. So we reanalyzed the male data in conjunction with the female data, and we are seeing that the females now really show, whereas the males and the males continue to show huge changes in this deep layer excitatory cell type, females are really showing interesting and vast changes in the microglial cell type and the inhibitory neurons. If you look at the bar graphs below these cluster differential expression profiles, you'll see that most of the changes that are occurring in the males are actually downregulation, and most of the changes that are occurring in females are upregulation. So that's interesting. So keeping in mind, again, these are the ones that are passing a threshold, not the sort of comparison across all changes, right? And the other thing is that most of the changes are unique. So that's what the other bar graph on the bottom is showing, is that most of these are actually unique to each cluster type. If we look at the microglia, this is where we're seeing the most changes in the females, and you can see that they do very much show a representation that is unique for the cases versus controls. And when we look at the Venn diagram, again, this sort of holds to the fact that those things that do pass threshold are quite different between males and females. So there isn't a lot of overlap between all the changes that we either see at the broad cluster level or even at the more fine cluster level. Most of those are unique to the females or the males. We also did a meta-analysis to compare the results of the previously generated male results and the female results. Because we generated the data differently, we couldn't analyze them together. We had to treat them as two different data sets because there would be too much of a batch effect. But we see by and large that this is consistent, that if you combine the data with a meta-analysis, you're usually increasing the signal in the same direction. But there are the rare cases where you see that the combined p-value actually is lower than one of the sexes, which suggests that the changes are occurring in opposite directions. But by and large, again, the combined p-value suggests that these changes are happening in the same direction. Again, we were able to find enrichment for depressive terminology in using Disgenet as an overlapping database. And when we look at the different networks that are related, we actually see this very interesting effect where the networks that are related to the microglia are networks that are more associated with neurons. But this is actually to be expected because these are the networks that actually communicate with neurons. So those networks tended to be upregulated in the microglia, whereas the immune signal tend to be decreased in the microglia. And again, all these changes are in the female context. For the inhibitory cell types, we were seeing a decrease in the stress response system and an increase in things that potentially recruit PNNs. When we looked at this in a kind of a different context, but we basically just wanted to evaluate the data in a complementary way to what we had done before. We used these network analyses where we see which groups of genes are regulated in the same way, and then we associate it to the phenotype. And so we do this WGCNA to do that. And what we see is that there is a particular module that is both associated with the phenotype, and of course, this is in the context of the microglia, again, in female. And we see that this particular module that has a related gene network has an enrichment for our DEGs. And if we look at what the ontologies, the biological functions, again, it goes back to neuronal systems. So this is going back to that first network that we had found just by looking at the DEGs. So it's just basically recapitulating our findings. And for we do the same thing with the inhibitory cells, and we see essentially the same thing, that we can recapitulate these findings. Finally, we did some network analyses, and we showed that certain networks are really more present in in cases versus controls, and that these networks are involved in the regulation of neuronal function by microglia. So it kind of goes back to this whole idea that microglia might be starting from a different set point, and that's why the microglial patterns look different in males and females. So I just have a summary statement here, but I'll read them quickly. While MDD is associated with differences that pass significance threshold with very little overlap between sexes, there is a markedly high concordance between the two sexes. Males show dysregulation in excitatory neurons. In contrast, females show a striking difference in their microglial populations. And then I just wanted to point out that these data sets can be merged in a meaningful way to derive more information. So thank you very much. Sorry for taking too much time.
Thanks a lot, Corinna, and thanks a lot, Chen, for the very nice presentation. It's really great to hear you. There are no questions on the chat, but I'm sure we can discuss together. So, well, of course, I would have many questions that would probably take 2 hours to to go through. But I guess perhaps we can start by trying to stay in what was our intention with the webinar, right? So perhaps the idea would be for you to give your impressions about the other speakers' presentation, and perhaps discuss about this idea of basically how many subjects do we need to analyze to, to saturate the understanding of the pathophysiology of depression, let's say. Easy question first.
Sure. I mean, I can start by saying that, while this webinar was, set up to sort of pin us against each other, I don't think that we're doing diametrically opposing things. I think that actually what we're doing is building data sets that can be merged, and that's kind of what I was showing there. So these findings with PRS and with epigenetics at the peripheral levels, where we're using larger population cohorts, are super informative. And then we can overlay that information in the data sets that I generated at a much higher resolution, so that we can see where these signals are coming from. So we have already started to try to do these things where we take some of the genetic components and we see how well they map to certain cell types, because they tend to be in genes that are more relevant to certain cell types. So I think that they're actually more complementary than this sort of what I was trying to say. I think they're extremely complementary, and that we can definitely leverage the information both ways. And I think that Chen would be able to use some of the single cell data that I generate to kind of query her data a little bit differently. Yeah.
I absolutely agree. And I think in an ideal world, we would like to have thousands of postmortem brains for each disease with all single cell analysis completed. But it's just, financially, it's not possible. So different people chose different routes. And then I guess what's a little bit more controversial is which part do you compromise? And I guess for Corinna's institute, they would emphasize on the depth of different types of cells they analyze, whereas in our institute, we're more focused on just finding the replications in the most accessible tissue type. And in fact, I really agree with the complementary side of things, because what we're doing right now is we're always struggling interpreting results coming from whole blood. And we would love to have information about epigenetic markers, about how consistent they are across different kinds of cell types. And as Karina mentioned, I think a lot of the large studies tend to give sort of markers so that they can conduct wet lab-based analysis. So we kind of need the information from wet lab single cell analysis across large number of different profiles, so that we can interpret our analysis in a more robust way. Yeah, but of course, yeah, people may have different prioritization strategies of which way you'd like to go first.
Yeah, no, absolutely, and perhaps to try to build on the two arguments that you've made. One of the things that I found striking in your presentation, Chen, is the when you mentioned the trans effects, right? So the notion that you might have SNPs at one place that seem to associate with DNA methylation levels at another place, and so I guess that could be one spot where, you know, trying to associate that with, for example, chromatin conformation capture data generated in the brain would be helpful in trying to make associations, you know, between distant loci, for example, right?
Yes, absolutely. And I think they're often, like you said, you very correctly pointed out some of the key genetic risk variants that have very global effects onto the entire methylome. There might be very key factors that pinpoints to the specific global biological process that influence, for example, histone modification, and those kind of biological process that can present in this way, and that shows a whole methylome effects. And certainly with our data, we aren't able to pinpoint what kind of biological process that leads to this kind of results. So absolutely, wet lab analysis would be super informative. I guess as a follow-up question or comment is that I guess it also depends on traits. And in my analysis, I particularly focused on MDD, but I noticed that in Karina's presentation, you focused on a lot of different disorders, and they may well have very, very different cell type specificities. Like schizophrenia and cocaine use, those kind of things may present very differently from depression. And from our experience, MDD is super polygenic compared to many other mental health issues. And they tend to be more heterogeneous and milder in many ways. So I think for MDD in particular, large samples will be really helpful, but maybe less of an issue for traits like schizophrenia and bipolar. Maybe less sample would achieve reliable results already.
Yeah. Perhaps Raoul or Boris would like to share some thoughts, or?
Yes, thanks a lot to both of you for this fantastic talk, very inspiring. Thanks a lot for that. And I have also the feeling that in terms of bench work, we have like a trade-off between exploring more and more CPG and also increasing the number of samples. Can you comment on that?
Can I ask the specifics, as in, are you suggesting since we increase the sample size, the end result is that we find more and more CPGs, or?
Well, the problem is that we have around like 20 million of CPG across the human genome, but it's really difficult to explore at the same time all the CPG. And for instance, the EPIC chip, which is the best tool for peripheral analysis, is less than 1 million. So yes. I have a feeling that sometimes we are missing a lot of information based on that. On the other point, when we use sequencing data coming from whole genome-based repeat sequencing, for instance, we cannot perfectly align the results for all the individuals of the cohort. So I think that there is a trade-off between more samples and more information about the CPG.
That's totally right. I think that's a really fair point. And that's exactly what we're finding sometimes, that a lot of the results are driven by MQTLs. And yet sometimes you don't find phenotypic associations, and our guess is that there could be true associations that is unmeasured. And what we're finding is just the results tagging along with the true associations. And I think that's very true. I guess it's kind of beyond my my ability to guess what's going to happen next. I know there are more like cheaper arrays developed, and there's I think it's called a 850 K array that is even better than Epig. And I think there might be a trend for DNA methylation data to be mapped more refinedly, just like the exome genomic data. But of course, that, again, it needs some sort of, actually, it needs a big sample as a reference data set so that we know how the CPG sites are correlated with each other. That helps future imputation, etc. So I think you're right that expanding the number of CPGs is really important. And actually the first step, in my opinion, might be just finding a really big, healthy sample, population-based sample, so that we know the methylome better.
Thanks a lot. And Corina, I was wondering if you have some clues about the single-cell methylation studies, because it seems that the field is emerging. I have the feeling that it's not totally mature yet.
So yeah, and I was going to comment a little bit on what Shen was saying too, because yes, we need to expand the number of CPGs that we can interrogate, but we also have to do this at a cell-specific level, because as you all know, the epigenetic profiles are cell-specific, right? And so this is always the real big challenge, is how well are what we see peripherally, are they corresponding to what's happening centrally? And it's a tough question to answer. And I don't think that that the information we're getting, from the periphery is invalidated. I think it's really important. I think it also can lead to biomarkers and other such things. But mechanistically, if we want to get into it, we need to really look at these things at the single cell level. So the studies are difficult because of many reasons, and largely because it's just extremely expensive. So if you think about it, it's the equivalent of doing whole genome sequencing for every single cell. So what I showed was 180,000 cells. That would be 180,000 genomes that we would have to sequence. Can we do this at the array level? It would still be 180,000 samples on an array. And that's for, across a few individuals. So it's a bit of an odd scale, right? Because you have the factor of an individual, but then you have a factor of how many cells do we take from that individual. And we can't just take one cell per cell type, right? So we do need a good population of them. So we're a little bit, there are some studies out, but we're a bit far away from being able to actually survey these things at the single cell level as of yet. But to Pierre-Eric's point, there are different ways to survey sort of epigenetic landscape, and that's using ATAC. And I think that's extremely complementary as well to the genetic variants that we see. It's really a good way to look at sort of trans effects as well.
Because it's cheaper, right? Mostly, that's the strong
It's cheaper,
argument.
because we can. So first of all, it's going to be only a portion of the DNA that's open and available for sequencing. And the methodology allows us to do it at the level of the single cell for less. Yeah.
And if I may, also about this issue of, or the question of where we're going in terms of DNA methylation analysis, I would say that similar to the whole sequencing field, right? I guess that the next big step is going to be switching to long-reads technologies, which should allow us, to analyze methylation patterns at the single molecule level across very broad genomic ranges, which is obviously going to increase the amount of data we generate by another factor 10 to the 10. But I guess that's probably a promising direction as well.
Absolutely. That's definitely the future of all sequencing, is moving into the long reads and sort of filling in the gaps for all the things that we were unable to sort of explore with the short reads. But to your point, it's another magnitude of data.
Yeah, perhaps Raoul has a question for.
Yes, if there is no question from the audience, but I guess there is no. This is more a comment than a question for Corinna, but in terms of pharmacological application, do you expect that we could have some pharmacological insight? Because in fact, the pharmacological treatment are acting in the whole brain and in several kind of cells. We several years ago talk about the fact that SSRI may act on astrocytes and etc. But how do you what do you think about the fact that we have some very waste and non-cellular specific treatment in one side, and we have a deeper knowledge at the cell level of the biological process?
Yeah, absolutely. I think this is part of the goal, right? So when I was saying this is complementary, it's, you know, some of the work that Chen does is to find biomarkers, but some of the work that I want to do with single cell stuff is to find novel targets, right? And so, yes, getting this precision and finding precisely which cells might be disrupted. And it's interesting because going back to this idea of heterogeneity in depression, there should be certain cluster symptoms that we can actually cluster individuals by and then find more personalized treatments. So individuals who have more metabolic syndromes that are associated with depression could be treated differently than people who have different symptoms, you know? And I think that looking at these data, this high resolution data in relation to the epidemiological data, putting it all together and finding different clusters can really help us personalize the medication and the direction that we go with targets. And I think that there are some interesting and novel therapeutic approaches, EVs, for example, where we might be able to direct our molecule to a specific cell type. So it's definitely opening the door to possibilities. And I think, you know, the ultimate goal here is to have a more directed and more specific approach to pharmacological treatment in the context of depression.
Just to support what Corinna said, I think absolutely, I think it's really helpful. The individual variability will be good for precision medicine, definitely. And I think that is sort of the ongoing research, maybe not happening for mental health disorders, but for cancer. So people are trying to do that. And of course, because cancer is a completely different kind of trait. And I think it just takes more time for mental health to catch on. But yeah, absolutely. I think those really high profile analysis will be super, super helpful to pinpoint the different symptom layout for different individuals.
Okay, great. So thank you very much. It's been really great to hear from you, and it's clear to me now that you know, with women in science, we have very friendly discussions and interactions that even when you try, they remain very friendly. And so thanks to both of you for that. And so I guess that's it for today. So Corinna, enjoy your class.
Thanks.
Thanks, Chen, a lot again. Thanks to everyone who attended the webinar, and see you for our next events, maybe.
All right. Bye, everyone.
Thank you.
Thank you.
Thank you. Bye-bye.
Goodbye.